بسم الله الرحمن الرحيم والصلاة والسلام على نبينا محمد وعلى آله وصحبه أجمعين. الأخوة والأخوات، السلام عليكم ورحمة الله وبركاته. Ladies and gentlemen, good afternoon. My name is Ghada, and today I will be taking you through the wonders of bacteriophages, inshallah, where I'll explain their history, structure, life cycles, bacterial resistance, and applications in medicine, food, and biotechnology. It is worth noting that this lecture is for educational purposes only. So some figures and data are adapted from published resources and used under fair use. Right, so let's begin. So in this lecture, as I have already mentioned, that we will go through history, early recognition, and then we will move on to taxonomy and classification. And then we will talk about the structure of bacteriophages, and we will talk a little bit about jumbophages. And then we will mention the life cycle of bacteriophages, and we will finish off with the resistance mechanisms and some applications of bacteriophages. Point 8 and 9 will be covered in separate talks, inshallah. Right. So when we say bacteriophages, one of the early or one of the first questions that come to your brain, who was the first person to actually recognize bacteriophages, or who was the first person to discover bacteriophages, and who actually named them bacteriophages, okay? So according to literature, someone called Frederick Tort, and that was back in 1915, was the first person to recognize non-pathogenic filter-passing viruses. Non-pathogenic filter-passing viruses. And you might be aware that filters in general are used to sterilize solutions and reagents in the lab, okay? And they stop bacteria from passing through. However, those viruses have the ability to pass through these filters, okay? So this scientist, or Frederick Tort, he is a bacteriologist in the 20th century. He's a British bacteriologist. He developed many things for the field of microbiology. For example, he developed Gram-Tort stain, which is different to the normal Gram stain. And he also characterized some bacterial species based on their phenotypes. He also developed a complex media for Mycobacteriaceae genus. So Frederick used to work in a laboratory in London, and if you look at the date here, or the year here, over here, let's put the laser pointing so we can, 1 second. Yeah, this one. So if you look at this year here, which is 1915, you would immediately realize that during this era, there was, the World War I was going on. So it had, of course, its effect on many things, including the scientific field. So he couldn't do any further work because of the work. So he wanted to do some cultivation and infection attempts, but he never actually continued his work on these filter-passing pathogens until 2 years later. Someone else came. He's a Canadian French scientist. This one here, Felix de Hydrel, and that was in 1917, during the war even. However, he came to the same conclusion. He noticed some filter-passing viruses. He was actually working on dysentery, and as you might already know, that dysentery is an infection caused by Shigella dysentery, okay? So he collected a stool sample, filtered the stool sample, and he took the filtrate, and he isolated the causative agent, which is Shigella dysentery, okay? And he did an infection. What he saw was what? Plaques formation. Plaques meaning the bacteria cannot grow in this clear zone, okay? So there was no growth, bacterial growth. So he came to the same conclusion as did the previous scientist, that these filter viruses are able to cause plaques and stop bacterial growth, okay? Just like the previous scientist, he also had some other contributions toward the field of microbiology, such as he isolated the infectious agent causing enteritis. For those viruses that pass the filter, he called them bacteriophages. Bacteriophages. Bacterio, i.e. bacteria, phages, eater. So those are viruses that eat or engulf bacteriophages, okay? And from that time on, he suggested that those bacteriophages should be used to treat bacterial infections, okay? However, bacteriophages had never been used to treat bacterial infections, especially with the rise of antibiotic industry. And there are a number of reasons as why people never used bacteriophages to treat bacterial infections, and they only rely on antibiotics. One of the very common or the very obvious reasons is that antibiotic is easily to produce, easily to be stored and transported, okay? It is not the same case with bacteriophages. They need special preparations, okay? So now let's move on to the taxonomy and classification of bacteriophages. And when we say taxonomy, we mean giving names to bacteriophages. Classifications, for example, classify them like in bacteriology, we go like a gram-positive bacteria and gram-negative bacteria based on their gram stain, okay? So what is the case in bacteriophages? Slightly different, but they are also being classified. Again, they have a history of classification, which I'll show in a moment. Well, before that, where do bacteriophages live? Well, they live everywhere. Anywhere on planet Earth, there are a number of bacteriophages. In fact, they encompass the most organisms on planet Earth. They came to over a count of 10 to the power of 31, and they prefer to live in the marine environments. However, they live everywhere. They live in sand, they live in soil, they live in worms, they live in compost, crops, in fresh water, salt water, extreme environments. In fact, and according to the literature, for each bacterial cell, there are at least 10 bacteriophages. For each bacterial cell, there are at least 10 bacteriophages. And there is a reason behind this, why bacteriophages outnumber bacteria, why bacteriophages outnumber any other living organism. Okay, so wherever there are some bacterial cells, there would be more number of bacteriophages, okay? So let's talk a little bit about their habitat. So if they live in ocean, which is marine waters, what kind of bacteriophages that live there? These are the, what is it here? Various tailed phages, and we shall explain what we mean by tailed phages later on, and cyanophages. What is their role? They regulate the bacterial population, influence carbon and nutrient cycles. So they have an influence on ecosystem. So if they live in fresh water, like rivers and lake, most of them are coliphage and cyanophages. Coliphage are bacteriophages that infect coliform bacteria, which is E. coli, okay? So you would assume that these bacteriophages will be an indicator of pollution, okay, or indicator of fecal contamination. And they also control bacterial population in fresh water. In soil, rhizophyre, the type of phages that live there are tailed phages and jumbo phages. Jumbo phages, just to give you a sneak peek, these are very huge type of bacteriophages. And they affect soil, those bacteriophages affect soil microbial dynamics and influence plant-associated bacteria, right? Plant surfaces or phyllosphere, which is the leaf actually, there are some bacteriophages that live there, and those that live there are classified as jumbo phages. They have some possible control of epiphytic bacteria. Epiphytic bacteria means bacteria that live on the leaves, okay? Animal, such as human gut, have we got some bacteriophages that live inside our bodies? The answer is, of course, yes. Okay, so what is their function or their main role? So they modulate microbiota, affect bacterial diversity. Okay. Combust or organic matter, the type of phages that live there are jumbo phages, and they may infect soil and decomposer bacteria. And then extreme environments such as sea, ice, and sediments, the type of bacteriophages that live there are mainly tailed phages or jumbo phages. They survive harsh conditions. They maintain microbial community structure. Last but not least, in wastewater like sewage, can we actually isolate some bacteriophages from there? Of course we can. Most of the bacteriophages isolated from there are coliphages, and they can be a good indicator of bacterial population. So what we understand from this table, that every niche, every environment, in every niche and in every environment, wherever there are some types of bacteriophages that adapt to live in to infect bacteria in that designated environment. And their main function, if you look at this column over here, their main function or role is to control bacterial population. So to sum up this table, we can actually say that bacteriophages control bacterial population. Bacteriophages control bacterial population, right? So why do we need to classify bacteriophages? Well, the answer is easy. For simplicity, we go tailed phages, non-tailed phages, or we go double-stranded DNA phages, single-stranded DNA phages, and so on. They also facilitate comparisons. They give us a better understanding of viruses, and we obviously use them for teaching, and we might be able to identify novel therapeutic phages, and of course to control the accuracy of data. So now let's give a little bit of history about the taxonomy and classification of bacteriophages. Even though bacteriophages were discovered, were named, were slightly characterized in 1917 by Felix de Hedrel, they weren't classified or taxonomized until many years later. So in 1937, bacteriophages were classified according to their size and resistance patterns to physiochemical agents. So you can see here, only in 1337, people started to classify bacteriophages. In 1944, bacteriophages were also classified based on their morphological differences. Morphological differences, how did they manage to to do this by use of EM. EM stands here for electron microscopy. So electron microscopy, even though it is a very ancient technology used to classify bacteriophages, is up to date used, okay? Until nowadays, people still realize on electron microscopy for classification of bacteriophages. In 48, bacteriophages were classified according to their host range and diseases caused. For example, those bacteriophages cause this kind of infection, whereas those bacteriophages cause other infections or other problems, okay? In 62, they were classified according to their properties and NA contents. What do we mean by NA contents? It simply means nucleic acid contents, whether they contain DNA or RNA, whether the DNA content is double-stranded or single-stranded, and the same strategy for RNA. Right. So in 1665, only very recent, ICTV was formed, and ICTV stands for International Committee for Taxonomy of Viruses, and this is the only committee responsible for taxonomy and classification of viruses, okay? Or nomenclature. In 67, people also classified bacteriophages according to morphology. So in 44, we used electron microscopy to classify them according to morphology. Further classification based on morphology were used in 67, were applied, sorry, in 67. And they looked at the tail structure of bacteriophages, whether those tails are contractile or non-contractile tails. So we could also classify bacteriophages as tailed, non-tailed. Tailed bacteriophages could further be classified as contractile tailed bacteriophages, non-contractile tailed bacteriophages, okay? And then in 71, well, it was formed in 65, but until 71, it was the only organization or committee for classification of bacteriophages, okay? So in 74, Ackerman and other scientists improved the classification of bacteriophages. In 81, 84, you see those names: Mayoviridi, Podoviridi, Cephuviridae. All of them are tailed phages, DNA, containing DNA as a nucleic acid. However, in this time, their names were introduced, okay? From now on, you see other bacteriophages, like in 78, other bacteriophages were introduced, and the name of those bacteriophages, like Enoviridae, Microviridae, Tectiviridae, Corticoviridae, and so on, those names were introduced and then were agreed on by votes, okay? In 98, Ackerman, he classified tailed bacteriophages. Those, the Mayoviridi, Podo, and Cephuviridae, were classified as codovirales, okay? From 18 and until 2020, honestly, until nowadays, classification of bacteriophages were based on gene sequencing, okay? Right. So we could understand that bacteriophages are classified based on several factors, based on host range. Most bacteriophages are species specific. What do we mean by that? Let's take, for example, Salmonella genus, Salmonella typhi species, Salmonella paratyphi species. So we have bacteriophages that attack Salmonella typhi only, and they do not attack Salmonella paratyphi. So they are species specific. According to nucleic acid type, so some bacteriophages contain DNA, other contain RNA, whether single or double stranded. Type of phage, is it lytic or is it lysogenic? Does it cause the cell lysis, or does it integrate and replicate with the cell replicating without lysing the cell? So is it lytic or lysogenic? Again, this is another way of classifying bacteriophages. Morphology, and what do we mean by that? Shape, icosahedral, does it have a tail or not, and so on. Genome size and replication strategy. What application strategy could be fused to this one here, type of phage, lytic or lysogenic? Genome size, bacteriophages of smaller genome are classified as bacteriophages. Bacteriophages of larger genome have a different name, which is jumbophages. Bacteriophages with extra large genome size, they have other name like megaphages, okay? So let's take a look at this information on this slide. So as I have already said that electron microscopy is used to classify bacteriophages. And according to electron microscopy, over 96% of bacteriophages are what? As you can see here, are tailed and contain double-stranded DNA. So most of bacteriophages are tailed and contain double-stranded DNA. So if you run an experiment and you would like to isolate some bacteriophages from whatever source of environment you are taking your sample from, you would expect that to a higher percentage that the bacteriophages that live in this sample, that are present in this sample are tailed and contain double-stranded DNA. For example, we have here Mayoviridis, Cephuviridae, and Podoviridae. These are the most common isolated bacteriophages. Mayoviridis account for 24.5%. Cephuviridae, the most common, account for 61%, and Podoviridae 14%. And those are families. So Mayoviridis is a family, Cephuviridae family, Podoviridae family. What about the rest? What about the 4%? Mostly are polyhedral, filamentous, like a filament, and pleomorphic, so they have different shapes. So let's take a look here. So tailed, we have Mayo, Cepho, and Podo, polyhedral, Corticoviridae, Tectiviridae, Microviridae, and so on. Filamentous, we have Enoviridae, and pleomorphic, we have, for example, Plasmaviridae. All right, okay. So bacteriophages, as I have already mentioned, are classified based on a number of things, one of which is based on their nucleic acid. Let's take a look at double-stranded DNA. So عندنا هنا Mayoviridis, Cephuviridae, Podoviridae, Cortico, and Tectiviridae. And those are double-stranded DNA phages. Single-stranded phages, such as Microviridae and Enoviridae. RNA phages, and whether single or double-stranded RNA, such as Cystoviridae and Leviviridae. Okay, so let's take a quick look at this table. So here we have DNA viruses, and over here we have the RNA viruses. DNA viruses, as I have already mentioned, could be double-stranded DNA or single-stranded DNA. The same scenario is here for RNA viruses, could either be double-stranded RNA or single-stranded RNA. Let's take one example from the double-stranded RNA. We have here Mayoviridis, family Mayoviridis. An example of this is T4. T4 is a classical example of bacteriophages that infect E. coli. Okay, so what do we have here? So they contain linear DNA. They contain a tail, and this tail is contractile. Okay, let's take another example. We have here Cephuviridae. For example, lambdaphage, which is one of the very earliest bacteriophages identified and commonly used in molecular microbiology methods. Their DNA is linear, long, and non-contractile tail. So you see the comparison. So over here is a linear DNA, the same thing. Over here is a linear DNA. Contractile tail, however, here is a non-contractile tail. So you see now the differences between bacteriophages. Let's take a look at single-stranded DNA. So we have here Microviridae. A very classical example is, you see this sign, it is not phi. It looks like phi symbol, but it is not phi. Whenever you see this symbol for bacteriophage, it simply means phage. Okay, it simply means phage. So phage X174 contains circular DNA, so it's not linear, it's circular, and conspicuous capsomer. Conspicuous capsomer. What do we mean by that? When you look at the capsomer, capsomer is the head of the bacteriophage. It's very obvious. It's very clear. It's under electron microscopy. Okay. Now let's move on to RNA viruses. We have here Cystoviridae, which contain double-stranded RNA, Phi6. This is a linear RNA segmented envelope and Yeah, right. So let's explain this. Most bacteriophages capsid contain only protein capsid. However, some bacteriophages contain an extra layer over the protein capsid, and this layer is composed of lipids, which is the same case here in cystoviridis. Okay? Single-stranded RNA, we have here leviviridis. An example is MS2. Again, it's a linear RNA, poliovirus-like. So throughout this lecture, we will come across some sentences where the literature says that bacteriophages have homology to eukaryotic viruses, have homology, for example, to poliovirus, have homology to adenoviruses. There are a number of reasons, one of which is evolution, maybe. Anyway, now, if we look at some pictures of bacteriophages, you see how different they look. Let's look at the double-stranded DNA. So in literature, in books, we only see those, okay? Maybe because those are, these are the most common. However, there are different types of bacteriophages, shapes, and all of these contain double-stranded DNA. Single-stranded DNA as such, and then the RNA viruses, and whether single or double-stranded. So what we can understand from this slide is that bacteriophages come in different shapes and different sizes, okay? Fine. Now, let's classify. We also said that bacteriophages are classified based on nucleic acid. Nucleic acid size. And I have briefly mentioned that normal phages contain smaller genome size, small genome size of less than 200 kb. So this is the norm. This is the classical thing. This is what you would expect when you isolate bacteriophages. However, tiny amount or certain amount of bacteriophages contain larger genome size over 200 kilobase pair. And those are classified as what? Jumbo phages. And now let's look at the differences between normal phages and jumbo phages. So here it says majority of, they encompasses the majority of non-phages. So most of bacteriophages are, contain small genome size. Only, until the time of the publication of this information, or just over 90 or just about 93 of bacteriophages contain larger genome size, and they are classified as jumbo phages. The genome in normal bacteriophages are modular and clustered, whereas in jumbo phages are scattered and non-clustered. So the genes in normal phages are clustered and modular. So we have bits that deal with replication, bits that deal with, for example, encoding proteolysis or whatever, and they are clustered closely to each other. This scenario doesn't happen in jumbo phages. The genes are not arranged in cluster, and they are scattered throughout the genome. So the genes are grouped into functional modules, like these genes are responsible for replications. They sit together, to put it simple, others for morphogenesis, others for lysis. It is not the same case for jumbo phages, where genes are distributed throughout the genome without clear modular organization. Having the genes arranged in clustered and modular make these phages very efficient, very streamlined replication cycle, strong host specificity, which is not the case for jumbo phages, where they go through complex replication strategies, and they have their own proteins and enzymes, as we shall explore later in this talk. Okay. So the evolution in normal phages are modular and horizontal gene transfer. Horizontal gene transfer simply mean acquiring foreign DNA and integrate that foreign DNA into the genome, or could be in a plasmid, and so on. However, the evolution in phages and jumbo phages are independently and suggest higher genomic plasticity. So they are very flexible, the genome of jumbo phages. Right. So now let's move onto the structure of bacteriophages. As we have already said, that these are bacterial viruses. When I say bacterial viruses, so you would immediately think that these bacteriophages infect bacterial cells. They do not act onto human cells. Okay, they might be, but normally they infect only bacterial cells. Okay, the structure and morphology of bacteriophages serve their role and benefits, and they need the bacterial host to exploit their machinery and propagate, so they cannot replicate on their own. So you would argue, are these living organism or are these non-living organism? Okay, you would simply ask this question. In the environment, they are not. Okay, but once they infect the bacterial cell, they turn into a living stuff. Okay. Some bacteriophages have a very simple structure, whereas others are complex. Basically, they are formed of a head and a tail. Okay, let's now take a look. So this is a classical bacteriophage example. You would assume that this bacteriophage belongs to Meoviridae family. It's composed of a head part, okay, and a tail. A head and a tail. The head contains at least two things: the capsid, which is icosahedral, formed of protein. The chemical structure is protein, and nucleic acid, whether DNA or RNA. Okay. And then we will move on to the tail. The tail is a tube structure, proteins, okay, tube structures, sometimes surrounded by a sheath, okay, and it ends with spike protein or base plate protein. From the base plate protein, other structure extend from this spike protein, such as spike protein, okay, and tail fibers. So the long legs here are tail fibers, and the short ones are spike protein, and those are the attachment stuff of the bacteriophages to the host cell or to the bacterial cell. Fine. Okay. So what connect the head part to the tail are connector protein. This one here in yellow, the collar and the whisker. Those are connecting protein that connects the head to the tail. Right. So the head, as I have already said, is icosahedral. It has got some homology of major capsid protein in adenoviruses. It contains genetic material, DNA or RNA, whether double-stranded or single-stranded. It also contains tails, whether some bacteriophages contain tails, other non-tailed. طيب, how can the bacteriophages deliver its nucleic acid from the capsid to the host cell if it doesn't contain a tail? Well, it does something called a proteolipid tube projection, which pierces the host envelope, and then it manages to eject its nucleic acid. Right. Connector protein, those are protein. They form alpha helix. The N-terminus of those protein forms alpha helix. Alpha helix is a simple organization of proteins. And those are responsible for the interaction with the phage inner membrane. So these interact with the phage membrane from the inside, okay? Joined to the head part or to another connection protein. Chimeric ring, they come from different sources, and they also have homologous that are present in contractile or non-contractile tails, okay? Receptor binding proteins, simply those structure, located at the end of the tail or the base plate, short filaments with bulbous tips, resembling an umbrella. Other structure, we have here spike protein. In fact, those tail fiber, those long legs are used for attachment, initial attachment, and to the host cell. And this attachment is reversible. The bacteriophages could detach at any moment. However, after this reversible attachment comes the irreversible attachment, and it is facilitated by those smaller ones, okay, by the spike protein. At this stage, the attachment is irreversible. The bacteriophage then starts to eject its nucleic acid to the bacterial cell, okay? Let's take some example. We have here Meoviridae. So what does it contain? It does contain a tail. This one here is a tail tube surrounded by sheath What is the function of this sheath? It helps in contraction of the tail and efficient delivering of the nucleic acid. Okay. Also, the base plate structure is present in Neoviridae. Acromanoviridi is very similar structure to the Neoviridae. However, it does not contain a sheath. Sorry, it however contain different structure here at the tip, star-like structure or three-fold prong-like structure. Or to make it simpler here, fork. This structure looks like a fork, and these are responsible for the attachment to the host cell. Cephuviridae, again, those are tailed DNA phages. The tail, however, is very long. It doesn't contract. So this is a non-contractile tail phage. It also contains a base plate. The tail is a central tape measure of protein which is bounded by a tail tube. So we have هو sheath. In Neoviridae, here we have tail tube. Okay? This tail tube terminates with the terminator protein. The base plate structure is present in Cephuviridae. Poduviridae, the very cute, the very tiny, the very small ones, they contain short and non-contractile tail, as you can see here. It is simply an extension which looks like a tube. This tube penetrates host membrane and used to deliver the nucleic acid. The connector protein, similar structure to Cephuviridae, and they pose no sequence similarity, however. So structurally similar, sequence similarity, there is no sequence similarity, okay? So all of the previously mentioned phages are double-stranded phages. اللي هو DNA double-stranded phages. Corticoviridae and Tactiviridae, as you can see, those are non-tailed phages. Okay, non-tailed phages. Their head is icosahedral. They contain some extra layer. Those structure, okay, or lipid layer. So how do these structure transform or deliver their genetic materials? Again, through a lipidic tube that pierces the host envelope. Nucleic acid, they could be either linear or circular. They also contain a lipidic membrane. Major capsid protein is trimeric and composed of a double beta barrel structure. Okay, corticoviridae and tactiviridae again. The major capsid protein of this example is an alpha helix, interacts directly with the phage inner membrane. They pose structural homologies to adenoviruses. RBP stands for receptor binding protein. Receptor binding proteins. Where do they present? They present at the icosahedral head. And they have homology to the N-terminal domains with human adenoviruses. So those structures are used for the attachment to the host cell, okay? Because they do not have a tail. Plasmaviridae, okay? You would simply imagine that they do not contain a capsid protein, okay? Just a lipid protein membrane. It's a simple membrane which contain circular genomic DNA. So no capsid. Genomes is contained in a proteinaceous lipid vesicle. Microviridae, again, those are non-tailed phages. They contain single-stranded DNA and several proteins. Some of these proteins are responsible for attachment. Some of these are responsible for fusion and so on. The capsid is formed of beta barrel structure. The DNA delivery happens through protein oligomerization and tube formation. Okay, DNA cross and joins the bacterial membranes, result in pi passing the periplasmic area. As you might be aware, the periplasmic area is an essential structure in the cell wall of bacterial cell. Okay. Anoviridae, okay, they have a very different morphology and lifestyle. These are formed of single-stranded DNA surrounded by 100 or 1,000 copies of major capsid proteins. The major capsid protein is a long helix. The N-terminal is a signal peptide for membrane translocation and is cleaved prior to protein assembly. So if we look at G3P, this is a tail fiber responsible for the host attachment. For example, okay. Cystoveride contains double-stranded RNA, non-tailed, and no capsid. Lipidic contains an extra layer composed of lipid. This lipid layer surrounds the icosahedral capsid. Okay, Leviviridae, they contain single-stranded RNA and two other proteins. The major capsid protein is the shell itself, or the head itself, and other protein, which is the maturation protein, the major capsid protein and the maturation protein, okay? The maturation protein is responsible for the interaction with the genomic RNA. This interaction happens during the interaction of the bacteriophage and the host cell. This maturation protein also serves as a receptor for absorption, okay? Right. So now we have covered most of the bacteriophages structure, and we have learned so far that they are different shapes, structures, and so on, okay? Let's take jumbophages. There is a reason why I chose jumbophages in this talk, which we will know in further presentations, inshallah. So jumbophages, as I have already mentioned, they have a very large genome, over 200 kb, in comparison to normal classical bacteriophages. Okay, they live in several habitats, just like any other phage: aquatic environment, soil, plant, some insects, fertilizers, animal excretes, and so on. Okay, so most of bacteriophages were isolated from gram-negative bacteria. Okay, over 95% of bacteriophages have been isolated from gram-negative bacteria. Only 4 isolated from gram-positive. Okay, so those are inhabitants of gram-negative rather than gram-positive bacteria. They have a very large virion size. The replication is slightly different. They encode their replication enzymes, transcription, translation, and nucleotide metabolism enzymes as well. Okay, so normal bacteriophage, when they infect, they only contain their capsid protein and DNA, or DNA. And they use the bacterial host enzymes like polymerases, RNA polymerase, DNA polymerase, and so on. So they are 100% dependent on the host cell. The case is not the same in jumbophages, where they come with their own replicative, their own transcriptive, their own translational enzymes. So they are slightly independent. They only need a host where they can replicate inside, because they cannot replicate outside. Okay? They also come with their degradative enzymes to break down the peptidoglycan layer. This feature makes them very flexible. They can infect a broader range of bacterial cells. Okay. So as I have already said, they come with their DNA polymerases, RNA polymerases, endolysins, glycoside hydrolase, chitinase, and more than one copy of tRNA or tRNA. Okay. I think I have got a figure showing how they replicate. Okay, after this one. So let's take an example here, phage G, just to give you a glimpse of how big they are. So they have a very big capsid that comes to a size of 160 nanometer, very long tail, 453 nanometer, and very huge genome, 497 kb, and they come with some other enzyme, okay? This isn't the normal case in normal bacteriophages. Only in jumbophages, they come with bigger head, longer tail, and bigger genomes, okay? And they contain their own replicative, their own enzymes, okay? So suppose we have this bacteriophage infecting for example, an E. coli. So it does that by ejecting its nucleic acid and some other proteins. For example, viron RNA polymerase, messenger RNA, shell protein, and fuzz-like protein. So this is the bacterial cell, okay? And this is the bacterial chromosome. And all of these stuff have been injected by the bacteriophage. Okay? So the fuzz protein extend to form a treadmill or to form a thread-like structure. And this thread is located at both poles of the bacterial cell, okay? Shell protein, when it's fully expressed, it forms a nucleus-like structure. Why? Because the DNA replication and the DNA synthesis happens inside this shell. And why is that? Because the bacterial cell has got its own defense system. And this acts like a shield, so as a protection for the phage DNA, okay? And what can you see here? What can you say about the bacterial chromosome? It's been lost or degraded. So at this stage, we can say host take over, okay? What happens in the next stage is bulge formation. The bacterial cell is swelled in the middle. The treadmill or the thread is elongated, and the shell is bigger now, contains many copies of the phage capsid and the phage DNA. In fact, these threads, they act like a treadmill. So they move, they push the nucleus-like structure toward the middle of the cell, toward the middle of the cell. Okay. Once the capsid protein is formed, they get encapsulated at this stage. So capsid proteins are moved and encapsulated with the nucleic acid and assembled with the tailed and spike proteins. This process continues. So you see here, capsid protein are encapsulated and fully assembled. This process continues until the number of newly generated or newly propagated viruses exceeds the bacterial cell capacity, and the bacteria will eventually lyse, releasing those bacteriophages into the matrix, and another cycle starts over again. This doesn't happen in normal phages. Okay, normal phages do not go through this. Okay, so these phages are obligate intracellular parasite, i.e. requiring a host cell. They manipulate and exploit the cellular machinery. They terminate the biological processes required for bacterial growth and shift those processes to propagate themselves. Okay? So types of bacterial life cycle, we have lytic, lysogenic, transition, pseudolysogeny, phage carrier state, and chronic infections in the case of filamentous bacteriophages. For the sake of today's talk, we will only cover the first two bacteriophages, okay? So lytic, if we look at this diagram over here, starts with the attachment of the phage particle to the cell, injection, and then followed by injection of genetic material, host cell takeover, and then the replication takes place until the capacity is exceeded, and then the cell will eventually lyse, releasing bacteriophages into the environment. So it starts with attachment, nucleic acid injection, phage synthesis, phage assembly, and then newly produces phages exceed the bacterial capacity, which leads to bursting of the cell. This is classical example of bacteriophages replication inside the host cell, and this type of cycle is called lytic. Results in lysis of the bacterial cell. Lytic phage or virulent phage. Okay. Phage facilitator factors. Those are factors that help the phage in achieving its goal, which is propagating. We have the fiber protein, which facilitate attachment. Phage enzyme that rupture the bacterial membrane and cause it to lyse or burst. What are those receptors that are being recognized by bacteriophages? We have OM, which stands for outer membrane protein. So those from 1 to 6, those are host cell receptors, i.e. present on the bacterial cell. Present on the bacterial cells. The bacteriophages recognize those receptors and attaches to them, okay? عندنا هنا outer membrane proteins, several other proteins, lipopolysaccharide, teichoic acid, lipoteichoic acid, or flagella. Okay. طيب. So the other type of life cycle is lysogenic or temperate phase. Lysogenic or temperate phase. In this case, it starts with a similar scenario to the lytic phase. Starts with the attachment, okay? Ejection of nucleic acid. The nucleic acid, okay, doesn't replicate independently. The host genome is not degraded or anything. The actual invading foreign material is integrated into the host genome. And the cell replicates normally, okay? The cell does replicate, and as well as the genomic DNA, and the genomic DNA contains both the host and the invader DNA, okay? So the chromosome here integrates actually into the host chromosome. This chromosome integration gives the DNA another name. Now it is called prophage. Prophage. And the cell containing this thing is called lysogen. A host cell now is named as a lysogen. Okay? Here it says during host cell replication, the bacteriophage genome also replicates. So new progenies contain both host cell chromosome and bacteriophage genome. So the life cycle starts with injection and integration, phage DNA inserted and becomes a prophage. Latency and replication, prophage replicates with the host. The host now is named as a lysogen. Induction, what do we mean by induction? In some cases, this lysogeny could turn into lytic. So we could exceed the genomic DNA of the phage, and it replicates independently of the host cell and result in cell lysis. How do we control this? How do we stop lytic phage, and how we make it only a temperate phage? By some repressor protein. So repressor proteins maintain lysogeny. So lytic, we have a free genomic DNA that replicates independently. In lysogenic, the DNA is integrated into host, and it is named as a prophage. In lytic, the host cell eventually is lysed. In lysogenic, the host cell survives and divides. The lytic bacteriophage is only known as bacteriophage. In lysogeny, the prophage DNA is named as a prophage, and the host is named as a lysogen. Okay? So here says some temperate phages transition into lytic phages and vice versa. When? When do you think? So in this scenario, we have the cell the bacteriophages attach, inject its nucleic acid, start to replicate independently and cause lysis, okay? Some phages, however, integrate and replicate with the host cell chromosome. However, in certain condition, this lysogenic life cycle could turn into lytic. For example, at the availability of nutrition, the concentration of salt, the oxygen level, presence of radiation, temperature, presence of heavy metals, environmental pollutants, super infections, or host density. To make this short, if the environment is favorable, there are more nutrients, okay? The lytic cycle is favored, result in lysis of the host cell. No nutrition, no favorable environment, harsh conditions, the lysogenic life cycle is favored in this case, okay? So now let's come to the last part of today's talk, which concern the resistance mechanism of bacteria to bacteriophages. So bacteriophages replication inside the hosts and inside the bacterial host is a multi-step process, as we have already explained. The bacteria could evolve through each of these steps and become resistant to bacteriophage infection. What do we mean by that? We said that bacteriophage recognize a receptor. If we modify this receptor, if the bacterial cell managed to modify this receptor, the bacteriophage can no longer bind to the bacterial host, so blocking of the bacteriophage from attaching and gaining access to the cell. Suppose the bacteriophage manages to attach and eject its nucleic acid, there are some defense mechanisms that break down these nucleic acid. Okay, if the nucleic acid is not degraded, we also could stop the transcription and translation. Okay, host cell takeover could be interrupted as well. Capsid formation could also be interrupted. Let's take a deep look at this mechanism. So let's look at example A. So this is the normal receptor, which is located on the cell membrane and is recognized by the bacteriophage. The bacteriophage attaches and releases its nucleic acid. How the bacterial cell deal with that? So it doesn't want this receptor anymore. So it does some genetic modification, okay, by mutation. This mutation will result in expression of a different receptor, or even loss or down regulation of the receptor. So the phage can no longer bind to its receptor. This is a very simple mechanism. Or production of a masking protein. Production of a masking protein that masks the receptor so the bacteriophage can bind to the bacterial cell. Or production of a decoy receptor. Decoy receptor. These vesicles are known as outer membrane vesicles. The bacterial cell cuts some bits of its cell membrane. This cell membrane contains the receptor. So the bacteriophage mistaken this outer membrane vesicle as the host cell. Okay. So production of outer membrane vesicle. I mean, if you when you study bacteriophage's life cycle, you would see a lot of production of outer membrane vesicle. Fine. Some bacteria will produce extracellular matrix or extracellular matrix. This matrix hides the receptor. So the bacteriophage cannot recognize this receptor. What is the formation? What is the composition of this matrix? Well, this matrix contains sugar or polysaccharide, lipids, proteins, and some DNA, all of which help in hiding the receptor. So the bacteriophage cannot recognize this receptor no more. Molecular method, for example, site-specific recombination, for example, shifting the promoter from one site to another site. This will result in switching on or off. Switching on or off, okay? And this results in no expression of the receptor. No expression of receptor. Or through this mechanism is called site-specific recombination. Okay, this is a reversible mechanism. Under certain condition, the situation could be reversed, and the expression would be on, and the receptor would be expressed normally. Slipped strand mispairing. This basically means either insertion or deletion of a nucleotide. Insertion or a deletion of nucleotide will again switch on or off the expression of some of the receptor, okay? Or methyltransferase, epigenetic modification. This is a chemical modification of DNA without changing the sequence, okay? Without changing the sequence. When we methylate the DNA and we make them, we mark them as a host DNA, so any other invading DNA will be recognized as a foreign and then eventually cut, okay? This is a very simple example of how bacteria or how a bacterial cell evade a resistance mechanism. There are more complex or more advanced methods such as production, such as the RM system. The RM system stands for restriction modification, and it is composed of at least 2 enzymes. So in this case, the bacteriophages attach to its receptor and it starts to eject its nucleic acid. However, this nucleic acid could face the RM system. The RM system is composed of at least 2 proteins or two enzyme, one of which is the restriction endonucleases enzyme, which function is to cut. So it cuts any foreign DNA. طيب, it is an enzyme. Can it cut the host DNA? Well, the answer is no, because we have another enzyme, which is the methyl transferase enzyme here. It adds a methyl group to the host DNA. This will render protection from the restriction endonucleases. Okay, we have other system like here, DISARM, which stands for Defense Island System associated with restriction modification. So it contains both the methyl transferase enzyme and the helicase enzyme. And a helicase enzyme. This will stop the bacterial, this will cleave the bacteriophages DNA. We have another system, which is bacteriophage exclusion system. Bacteriophage exclusion system also contain a methyl transferase enzyme. However, it does not contain another enzyme that cleave the phage DNA. It only blocks the transcription, that's it. And then we have AGO, which stands for argonate. It contains nucleic acid complement. They bind to the complementary foreign DNA and cause cleavage. So all of these, so RM system, DISAR, and AGO, they cleave the DNA or the invading foreign material. However, PREX does not do that. CRISPR-Cas this is a very common or a very famous or familiar defense system. This is an innate defense system. So cluster regularly spaced palindromic resistance Cas protein. Again, it is formed of an immunity. So, for example, when bacteriophages invade the bacterial cell, bits of its DNA is saved in the bacterial genome in spaces. So whenever a second bacteriophage infections happen, those DNA are recognized, and the CRISPR encode for an enzyme that cleaves those foreign DNA materials. Okay, we have another method. Those methods are related to the cell suicide. We have CBAS theoresis. CBAS, which stands for oligonucleotide-based anti-signaling system. Those, they signal that there is a bacteriophage infection, and they cause cell lysis through the induction of enzymes. Some enzymes, those enzymes degrade the bacterial cell membrane and cause what? Cause cell death. Okay, cause cell death. Theoresis, again, senses the bacteriophage's infection and cause NAD depletion. Retrons, again, they bind to the foreign, they also cause, they also sense the bacteriophage's infection and lead to cell death. And a fourth method by the interruption of balances of toxin and antitoxin. So the biological processes that happen inside the bacterial cell produce some toxic product. Okay, so the bacteria deals with that by production of antitoxin to protect itself. However, when there is a bacteriophage infection, the balance of toxin and antitoxin is irregulated, where the toxin level exceeds the antitoxin, and this will eventually lead to cell suicide. Okay? We have some chemical defense, just some chemical compounds produced by the bacterial cell to degrade the bacteriophage's DNA, and we have some other uncharacterized defense system. Okay, so suppose the bacteriophages manage to infect a bacterial cell. Some type of bacteriophage infects a bacterial cell. It sounds horrible, okay, but sometimes it gives defense, it gives protection to this bacterial cell. Let me show you how. Okay, so this is the invading bacteriophage. It injects its nucleic acid, and it helps the bacterial cell by expression of at least 2 types of protein. One protein does mask the cell receptor, therefore stopping any other infection, any other bacteriophage from binding and infect. And production of membrane-associated protein, again, this will stop the bacteriophage from binding. So this is called protection caused by bacteriophages infection. So only one bacteriophage infects a bacterial cell. Other bacteriophages cannot infect the cell, okay? Via production of protein masking enzyme, or membrane-associated protein. And some molecular method that disturb the foreign DNA, such as RM system, or DNA binding repressor protein, or causing abortive infection. Abortive infection, for example, for this system here, when a bacteriophage, another bacteriophage start to infect, some sensors are located in the bacterial host. They cause depolarization. You see here plus and minus and so on. Depolarization of the cell, which will eventually lead to cell death. Okay. So these are how the bacterial cell evade, or how the bacterial cell protect itself from bacteriophages infection. Will bacteriophages be happy with that? Of course not. The bacteriophages will evolve counter mechanism to evade those resistant mechanisms, such as, so as the bacteria changes its receptor, the phage immediately introduces mutation in the protein that bind to the receptor, okay? Or bacteriophages could also bind to capsule. Capsule is not a normal, well, it is. If the receptor is not available for the bacteriophage to bind to, it can then bind to the capsule. Bacteriophages could also develop ways to evade the restriction modification system. CRISPR, as we said that some bits of foreign DNA is saved in the DNA of the host. Those DNA could also are called protospacer adjacent motif, could be mutated. So this way the bacteriophage will evade the CRISPR system. Abortive infection could be evaded by or inhibited by inhibiting the proteases enzyme. Again, mutations in genes involved with the metabolizing of genetic materials. Genes that activate the abortive infection of bacteriophages again go through mutations. So it is a balance. The bacterial host cell will develop resistance mechanism. The bacteriophage will develop evasion methods. Okay. So this leads us to the last part of my talk today, which is the applications of bacteriophages. So now we have gone through the structure, the function, the life cycle of bacteriophages, and the first scientist who named them bacteriophages actually suggested using them to treat bacterial infections. So the immediate application that would come to anyone's mind, why not use them in the medical field as treatment? Well, they could be an alternative way to treat bacterial infections, especially in the rise of bacterial resistance. However, this approach is still under development, and actually nobody is using, or on a global scale, no country is still using bacteriophages to treat bacterial infections, and it is still debatable, still under researches. People are still using antibiotics to treat bacterial infections regardless, except in some Eastern European countries, such as in places like Georgia, where therapy with bacteriophages is normally used. In places like the United Kingdom, bacteriophages is not used, and they still rely on the use of antibiotic. However, in certain severe cases, in urgent cases, when the patient is suffering from multi-drug resistant pathogen, a pathogen that doesn't respond to antimicrobial treatment, they would then and only make a custom-made bacteriophage for this specific case. okay, for a specific case, and only. I mean, you could come up with some reasons why people do not use bacteriophages until now, and why are they relying on only on antibiotics. And the simple explanation would be the easy production of antibiotics. They are very easily stored. They they get less contaminated than bacteriophages. If any one of you is familiar with bacteriophages work, you would you would realize, or you know that a stock preparation of bacteriophages needs to be filter sterilized every now and then, because they are very easily contaminated. Okay, so in a larger scale, you would imagine the level of contaminations that might happen. Okay, and one other reason why one could argue why people are still using antibiotics instead of bacteriophages is that bacteriophages, as we have already mentioned, are species specific. So a bacteriophage attacking Salmonella typhi is not attacking Salmonella paratyphi, for example. So you would produce a bacteriophage for each species. Okay, whereas antibiotics could act on both gram-negative and gram-positive, okay? Right. However, we could still use bacteriophages, especially during bacterial resistance. For example, in bacterial resistance against antibiotic, the bacteria, for example, will modify its receptor, modify its whatever. And then we could add the bacteriophage to the resistant pathogen. The bacteriophage will attack the resistant pathogen, will make it sensitive to the antibiotic. So resensitize the bacteria to the antibiotics, okay? So here is saying bacteria are made sensitive to antibiotic because of the action of bacteriophages. Changing receptor, bacteria become طبعا resistantly to antibiotics. Bacteriophages and antibiotic could have a synergistic effect on resistant strain. I'm not sure if you ever heard of this term before or not. The bacteriophages cocktail is a number of bacteriophages in one suspension or in one solution. Each have its own mode of action, okay? Right. When a species or more overgrows, it causes harm to human health. And this overgrowth normally or most of the time is caused by the administration of antibiotics, okay? For example, هنا عندنا overgrowth of Clostridium difficile. Clostridium difficile is present in the colon, okay? And it is suppressed. The Clostridium difficile is suppressed. Why? Because of the abundant microbiota, okay, in the gut. However, by using antibiotics, we eliminate this microbiota. The Clostridium difficile overgrow and cause some severe consequences. However, if we use bacteriophages to tackle Clostridium difficile on its own, it will leave the normal microbiota of the gut on its own without harm. Helicobacter pylori is involved in dysbiosis. As you might be aware, that Helicobacter pylori causes stomach infection, which ranges from normal gastritis until the very last stage, which is cancer, okay? So it causes gastritis, peptic ulcer, and cancer. So if you use antibiotic, as I have already said, some antibiotics have broader range, so it will affect the microbiome of the stomach. However, if we use a Helicobacter specific bacteriophage, it will only attack the Helicobacter bacteriophage, leaving the rest of the microbiome unaffected, okay? In food industry, in fact, bacteriophages could also be added to the pre-harvest and post-harvest. Pre-harvest to stop insect, to stop bacterial contamination of the crops, and post-harvest as a preservative. It will also lengthen the shelf life of the products, okay? So it minimizes the bacterial contamination of food products. As I have already said, lytic bacteriophages used in pre-harvest as well as in post-harvest. Working areas and surfaces could also be sprayed with bacteriophages, and this will eliminate food contamination. In fact, in hospital settings where biofilm production is a major concern hospitals' infection control, bacteriophages could also be added to the solution used to disinfect areas. It will help breaking down of the biofilms, okay? It also helps in controlling of pests and global warming. Okay, we shall finish off with this slide in here. So those insects here have some microbiome, okay? And these insects will damage the crops, okay? Will damage the crops because of the microbiome that lives inside those insects, okay? If we add bacteriophages, okay, bacteriophages will attack the microbiome of those insects and we save the crops, okay? Bacteriophages, if used in food industry as sprays, we will help in preserving and disinfecting of the food, okay? In combating global warming, this is interesting because in cattle, some forms of life like archaea and bacteria produce byproduct, and those byproducts will elevate the global warming via production of methane and carbon dioxide. And this all could be tackled by adding bacteriophages to the food administered to those cattle. Finally, in human health, bacteriophages could also help in treating dysbiosis, modulating the microbiomes, and it could also be used in non-bacterial infections. Okay, so this sums up my talk, my talk today. Here are some of my references that I have gathered the information from for today's talk. Again, this lecture was prepared and delivered for educational purposes. Figures and data are used under fair use and cited accordingly. Many thanks for your attention. And if there are any questions, I'll be happy to address and answer. Thank you. Are there any questions at all? الله يعافيكم أجمعين. شكرا لكم للحضور. الله يعافيكم. أتوقع إنها ساعة ونص المحاضرة. وإياكم. الله يعافيكم. أهلا وسهلا. الله يعافيكم. أهلا وسهلا. أهلا وسهلا. العفو. ما عندي فكرة صراحة الجواب هذا السؤال، هل هناك ساعات؟ ما عندي فكرة. بالنسبة لتسجيل المحاضرة بإمكانكم مراجعة ما أعرف، ما عندي فكرة للأمانة. المحاضرة نفسها ما أتوقع إنه أنا أقدر أعمل لها شير لأنه فيها some copyright materials، لكن راح أكون مرة مبسوطة إنه أنا أعطيكم آخر شريحتين تبعت الريفرنسز. الريفرنسز I can provide. العفو، حياكم الله. ما عندي فكرة عن الساعات للأمانة. طيب يا جماعة الخير، إذا ما في أي سؤال يعني يخص المحاضرة، حابين نقفل. يعطيكم العافية على الحضور، وأتمنى إنه فعلا أكون يعني قدرت أوصل لكم معلومات مهمة ومفيدة عن البكتيريوفاجز. السلام عليكم ورحمة الله وبركاته.